Okay, I'd like to start the second half of the morning session. I'd like to introduce Dr. Esther Schirrmacher. I worked with Esther off and on for a little while, and she's, as it states in her bio, Esther is an internationally recognized expert in regulatory affairs and GMP compliance for radiopharmaceuticals. And I can concur. She obtained her PhD in radiopharmaceutical sciences in Germany. She supervised radiopharmaceutical operations for commercial FDD production, as well as research radiotracer program at the McConnell Brain Imaging Center of the Montreal Neurological Institute for 5 years. She then assumed a position as regulatory expert at the University of Alberta, where I got to know Esther relatively well, where she was responsible for a clinical trial with technetium-99M produced on a cyclotron. Since 2019, she has been employed at Triumph as the Director of Operations for the Institute of Advanced Medical Isotopes. With that, Esther.
Hello, everyone, and good morning. Thank you very much for your kind words, Doug. A short disclaimer before I start. I have 45 minutes to tell you everything there is to know about regulatory and GMP aspects in radiopharmaceuticals, which is obviously not an achievable feat. So what I do hope to accomplish is to give you a basic concept and to provide links in the slides that will take you further for you to read up on particular aspects that are interesting to you. So without further ado, let me start. Okay, and now my slides don't advance. Okay, let me see. There we go. Okay, so there's two regulatory bodies in Canada that are really important on a federal level. The first, of course, is the Canadian Nuclear Safety Committee, the CNSC. They are concerned about protection of workers and or patients or subjects from the noxious effects of radioactivity on the human body. Health Canada, their mandate is obviously general health protection for the entire Canadian population. However, in the terms of, in the concept of radiopharmacy that we're talking about today, their concern is to protect the radiopharmaceutical drug product from the noxious effect of human beings. So it's a very complementary view set. And then, of course, there's further regulatory bodies that are not federal but provincial, and that depends on which province you're working at. This could be colleges that regulate certain things in pharmacy. This could be work-safe organizations that look after general work-safe conditions at your radiopharmacy. I will be concerned really about the Health Canada aspects today. So just quickly, the radiation safety aspects to get them out of the way, so to speak. In Canada, there are no dose rate limits for patients if they are treated under a physician's care with an approved radiopharmaceutical. And the reason for that is simple, that during the approval process, and I'll go to that in a moment, a risk-benefit analysis is actually already performed by the regulator, Health Canada, and so they say, yes, this is an acceptable radiation dose for the gain, whether therapeutic or diagnostic, that you get out of the use of that radiopharmaceutical. This is different, obviously, if you do a research study. So for any research study, the dose rate limits for study subject is regulated by Health Canada in the Food and Drug Act to 50 millisievert for the entirety of the study. So this could be either one single scan or therapeutic dose, if this is what your study is looking at, or it could be a longitudinal, several diagnostic and or therapeutic interventions. So now, how can you get access to a radiopharmaceutical and get it into a human being in Canada? There's basically 4 pathways. The routine, regular pathway that the regulator would like you to use is a radiopharmacy submits either a new drug submission or an abbreviated drug submission. That is really a generic, if you will. That's mostly used for FDG submissions. And then you have an approved marketed drug product, a radiopharmaceutical, that you can use either according to its label or also off-label if the physician so chooses. You also have the special access program, which I think Doug taught you something about, so I will not actually go into any details on that here. It is a vehicle to have 1 dose of the radiopharmaceutical for 1 identified specific patient. So the documentary and administrative overhead for that is huge, which is why it's not a dreadfully useful pathway. Other pathways are the clinical trial application, of course, and then what's called the BRAP. You see Health Canada is very fond of acronyms that don't sound overly great. It's short for the basic research application with positron-emitting radiopharmaceuticals. So before I launch into detail of all of those, I just want to let you know that there is a guidance document about the management of drug submissions and applications from Health Canada. It's fairly new, 2 years old. And it's actually a very, very helpful resource. It will give you clear instructions of how each of those pathways, how you can apply, what format you need to submit, what kind of information you need to submit, what standards they would ask you to respect, etc. So please use this as a roadmap if you ever find yourself in a position of wanting to go down regulatory route to have access to radiopharmaceutical in your life. So, which regulatory pathway of the 3, and I'll exclude the SAP here because, as I mentioned, it's just a one-off, should you choose? In theory, the answer is very clear. BRAP is to answer a research question that you cannot answer by using an approved drug in Canada. A CTA is really designed to develop a potential great radiopharmaceutical, a new one, towards its eventual commercialization. And the new drug submission, or abbreviated new drug submission, is really for commercialization. So you have a great radiopharmaceutical, you want to bring it to market, sell it, scan lots of patients, make it into a routine patient care thing. So let's go into a little bit more detail about each of those and look at the limitations and the strengths of each of those pathways. For BRAP, all you need is a clear research question. Again, the data that you generate cannot be used for the clinical care of the patient. So you can't really kind of use it to stratify patients into something. It's really used as a research tool only. The application is very, very easy to do. It does not require a lot of time. You have to submit a clinical plan, what you plan to do. And actually, you can really just use a grant application summary for CIHR, NIH, or something like that. You are limited to not more than 30 patients. There are possible exceptions. However, in my professional life, I have very, very rarely seen anyone who got approval to exceed 30 patients with a BRAP. And the drug data that you have to submit, so the manufacturing information about the radiopharmaceutical you're planning to use, is very, very limited. First of all, you have to reassure them it's just a checkbox that it has an established safety profile. And that means automatically that you cannot use a BRAP for a first-in-human radiopharmaceutical, okay? It has to have, someone has to have shown already that it is safe to use in humans. And then you just need to tell them what your maximum injected dose is, and that is cold and hot. So that is just to show that you're still in the tracer level and that there's no pharmacokinetic effect to be expected. They ask you about the excipients, ethanol, buffer, saline, whatever it is, the injected activity range that you want to do, and some dosimetry of it, and that's everything. So it's a very, let's say, a low regulatory hurdle to clear, which for Health Canada means that it's a high risk thing, and that's why they limit it to the number of 30 patients. So again, for the application, you submit the BRAP application that consists of information I've just told you about. You also need to already submit the Research Ethics Board approval. And that is because Health Canada does not make a judgment whether it's ethically justifiable to undertake that research. They outsource that to the REBs. And then a checklist, which is really just very quickly showing that you've met all of the conditions that make a BRAP permissible. Health Canada, on the other hand, then reviews and approve it within 15 days. So this is really quick. You can do great things with it. And they will then issue an authorization to sell letter to the manufacturer, meaning the radiopharmacy, that allows me to ship the product to the clinic where it can be used. In clinical trial applications, they are differently regulated in Canada, so they must follow the Division 5 of the Food and Drug Regulations. It is generally accepted that there are 3 phases of clinical trial applications. The first one is safety, of course, very limited number. So this is a possibility to do a first-in-human in Canada. The second one is some more safety data on a larger population, some dose finding that is not necessarily that important in the diagnostic radiopharmaceutical field, also some efficacy data, of course, and then phase 3 is just on a larger number of patients. Some say there is a 4th clinical trial application phase, and that is for the post-market safety, which means after you have filed an NDS, you need to still continue to monitor safety for any emerging super rare side effects or adverse events. So if you need to submit a clinical trial, it consists of 3 modules. The first module is administrative and clinical info. So that's basically who conducts the study, who's the doctor, how is informed consent obtained, who is the RAB, how are you scanning if it's a diagnostic, or how are you dosing, how much are you injecting, what are the time differences, etc., etc. Is there any other test that you're doing? And then module 2 and 3 are concerned with the radiopharmaceutical, the study. So module 2 is just a relatively short quality summary. Module 3 contains a heck of a lot more of additional quality information, and that one requires more information as you move from phase 1 to phase 3 of your clinical trial application. If you have one manufacturing site, so one radiopharmacy, but you want to have several clinical scanning sites, then you can also combine module 2 and module 3 into a drug master file that you file with Health Canada. And that has then the advantage that each clinical site just needs to submit their module 1, so you don't need to copy paste your entire module 2 and 3 for each subsequent application. And then the clinical site in their application for the CDA will just submit module 1 and will make a reference to the drug master file that the radiopharmacy has submitted. So what are the requirements? You submit the CDA package consisting of those 3 modules, either in the ECDD format or the non-ECDD. That depends very much. Health Canada is starting to not allow that many non-ECTD electronic only submissions. And this usually it's submitted by the sponsor, or if you go the drug master file route, it's submitted by the manufacturer, and the module 1 then by the clinical site. Health Canada will review and approve within 30 days. And if they send clarification questions, which they usually do, it's called the Clarifax because they're still using fax, then the clock stops until you've answered for the 30 days. They will then issue a no objection letter. And please note, this is not a yes, this is a neutral, we have no objection, right? Because they consider there is some risk and they don't really want to go out on to expose themselves to risk, I assume. And they will then also issue an authorization to sell letter to the manufacturer if this is different from the scanning side. Now for new drug submissions, the culmination point of your radiopharmaceutical drug development. You obviously have to submit a lot of clinical data, including proof of safety efficacy. So those are the outcomes from your clinical trial that you've done, and now you're moving to the new drug submission. In terms of radiopharmaceutical, of course, they are very interested in dosimetry. That is the main concern, and also the usual safety that is important for all of the drugs. The manufacturing data is very extensive, and it's basically supercharged CTA manufacturing data information. So they want to know about your process details, about the controls of your process that you have, including possible or frequent, or well, hopefully not frequent, but occurring failure points, risk assessment, what that means for the quality of your drug, etc., etc. They also want to have detailed information about your materials and supplies. So they want to know about the purity and also the impurity profile. So for example, if you're using acetonitrile to make FDG and it has a 99.9% purity, they want to know what the 0.1% impurity is and whether that can have a negative impact on the drug quality or the safety for the patient. They also want to see evidence that, for example, if you change supplier and it's still a 99.9% purity of acetonitrile, but you have a different 0.1% impurity, whether that makes a difference. So you need to have means and methods in place to capture that and assess the risk before you actually start using that new source of acetonitrile. Of course, they want to see lots of information about the product in terms of purity and impurities. That goes without saying. They want to know about what quality control you're doing, what specifications your product, your QC tests have, but also what testing methods you're using. And they want to see evidence that you have validated your process and your test methods. And in the GMP part of my talk, the second part, I'll tell you what test method validation actually means. So the process per se. Oops, sorry, I seem to have done something wrong. Screen sharing has now paused. Can someone tell me whether you still see my screen?
Yes, we do see it. It's on new drug submissions.
Okay, perfect. So I'll just ignore that warning. Great. So in the new drug submission, you submit your new drug submission or your ANDS, and you also have to submit a drug establishment license application, which is for the radiopharmaceutical production site, and it enables Health Canada to inspect you and check on you. So Health Canada then will review and approve, hopefully, your new drug submission within a year. So you see the review time is much longer than the CTA with 30 days. They will then issue a notice of compliance. So that's actually the thumbs up. That's no longer neutral. They'll say yes, because now they've done an in-depth review. They feel more certain about being able to endorse that. They will issue a drug identification number, which will allow everyone to identify your drug specifically. And they will also inspect the manufacturing sites or the radiopharmacy and issue, hopefully, a compliant rating for the drug establishment license. And that means you're GMP compliant. And I'll go into a little bit more detail. So what drugs are available? What approved radiopharmaceuticals are available? There is a drug product database by Health Canada. I did not include a link because they keep putting it in different places on their website, but if you just Google drug.
Esther, sorry, it's Mihaela. Your presentation is paused, so it's stuck on the new drug submission slide.
Oh, thank you very much. That was not supposed to happen. Let me go back on this. Okay. And let me go back to where I was here. And okay, I can't do it. I need to go through that. Okay, are we back?
Yes, now it's okay, thanks.
Awesome, perfect. Thank you very much for letting me know. Okay, so there is a new product database by Health Canada, so you can open that. Just Googling it, it will give you, it will bring you up to it, and you can actually select radiopharmaceuticals, and then you'll see exactly what radiopharmaceuticals are approved, pending, or lapsed in Canada. And there's a large number of SPECT product, and I'll not take away Dr. Fonge's thunder there. So you'll hear all about those in a moment. On the PET side of things, there is 1 or 2 gallium-68 generators. There's a gallium-68 labeled peptide. There's FDG and there's fluorobetaben, and that's it. Not very much, right? And I would like to remind you that Health Canada obliges you to use an approved product if it's available in your area over a research-grade radiopharmaceutical. So if you're sitting next to a radiopharmacy that sells FDG at a certain price, but they have an approved marketed product, you have to buy it through them. You can't do a CTA in your own institution with it, okay? So why do we have so few approved radiopharmaceuticals for PET in Canada? And the reason is very simple. Drug submissions are costly, and it's a cost versus expected revenue thing. So why are radiopharmaceuticals specific, or why do they have specific problems in Canada? First of all, they have a short shelf life, of course. Goes without saying with the physical half-life. Second of all, though, there are challenges because Canada is a geographically huge country and the population density is varied, but not super high. So out in Quebec and Ontario, the population density is fairly high, which is why you see a large number of approved FDG manufacturing sites there. And also fluorobetaben is available there under the Neuraceq trade name. Out west, where I'm sitting, I'm in Vancouver, we have a relatively low population density. So within an acceptable driving or even helicoptering distance, we have very few patients. And so it's just not worth the amount of burden and money to submit a new drug submission. It just doesn't happen. And of course, it doesn't help Canada that healthcare is provincially regulated and funded to a large extent. And that means that there are different rules and caps in each province, which makes it hard for a manufacturer to set up shop basically and become financially sustainable. So how does the practice of the regulatory pathway actually look? NDS, as we've seen, limited to very few. And that leads to the fact that many sites do what I call pseudo research, and I hope that Health Canada never sees those slides. What it means is that we disguise patient care as a research question so that we can file a CTA and give our patients access to a radiopharmaceutical. That is, I mean, no one really wants to do any more research on gallium-68 labeled PSMA or lutetium-177 labeled PSMA. That has been done, but we disguise it as research. We come up with hopefully relatively large sample size calculations. So we say we file a clinical trial application, we can do 300 patients with it, and then we do patient care and pretended we're doing research. PrEP is used in Canada quite a bit because it's such an easy access. However, it's usually used only by kind of really research centers that have the necessary critical mass in terms of researchers and radiopharmacy and grants to follow that pathway. So this here is just a kind of a summary slide. I won't go into detail, but what I would like to draw your attention on is which regulations you need to follow for which pathway, with a specific view, obviously, on GMP. So for BRAP, I put GMP light in quotation marks, and again, I hope Health Canada doesn't see it. It means if you're doing a radiopharmaceutical for a use in a BRAP, they expect you to meet GMP, but you can have some areas where you're not fully compliant. If you've done a proper risk assessment and it's a defensible thing, they will allow you to go ahead. On the CTA side, as already mentioned, the GMP compliance that Health Canada requires you to do is increasing from phase 1 to phase 3. So they want to see consecutively more areas covered that you need to cover. And then, of course, once you file an NDS, you have to definitely be 100% compliant to GMP. You also have to be compliant to GDP, so those are good distribution practices. That's how you ship your drug to the hospital or to the scanner. Good pharmacovigilance practices, that's what the V here stands for. So that is that kind of post-market thing that we're going, data integrity, et cetera, et cetera. So you can see as you move along that pathway, the GMP compliance and general regulatory compliance requests become higher, as they should. So now I want to go towards the GMP and tell you what is GMP, so you can understand what you need to do. The GMP regulations are enabled by the Food and Drug Act in the Division 2. They are always accessible for free. You just Google Health Canada Good Manufacturing Practices, or you follow the link on the slide. I believe you'll be given the slide deck after that. They're organized on Health Canada's website into kind of subject matter areas, if you will. So you have a section on good manufacturing practices, guidance on pharmacovigilance practices, on CTA for the manufacturing GMPs, like what are they looking out for specifically there. There's also guidance on compounding, establishment licenses, radiopharmaceuticals in general, etc. So which are important for a radiopharmacy? Of course, there's Guide 0001, which is a general GMP guideline. Most important, you need to familiarize yourself with it if you ever work in a radiopharmacy. It is not specific to radiopharmacies, it's for all drug manufacturers. Then there is Annex 1, also called 0119, and that is the manufacturer of sterile drugs, which most radiopharmaceuticals, and well, all of them if they are injected, which they usually are, need to follow as well. Then there is the Annex 3, which deals with Schedule C drugs. So in Health Canada speak, radiopharmaceuticals are called Schedule C drugs. So this is a specific one for radiopharmaceuticals that takes into account some of the specific nature and kind of like where radiopharmaceuticals are special as compared to the regular drugs. And then you have the Annex 3B, which is specific to positron-emitting radiopharmaceuticals, PERPs. So this is where the PET imaging falls under. And those guidelines are updated, I won't say on a super frequent basis, but on a regular basis. So please be aware that if you print out something or save it locally on your computer, you might work with an outdated version. Again, they have relatively recently published an excellent roadmap document, and I've included a link on the slide here that tells you really kind of in what type of submission they will expect you to follow which of the guidelines. Okay, and one thing to keep in mind is the guidance documents are guidance documents, and it bears saying that twice. So they tell you, they guide you to what they would like to see. You can always choose an alternate approach if you have a proper justification. That is what they put on the front page of all of those. That being said, usually when the inspectors come to inspect you, they really want to see you to follow those guidance documents, so it's less of a guidance. Other resources that Health Canada would like you to be aware of and follow to some extent are USP chapters, some specific ones for, say, FDG or something like that, so for monographs, or also some generic ones about microbiology testing, etc., etc. There are also ICH guidelines, so that's the International Conference on Harmonization. Health Canada is very big on harmonizing what we in Canada do as compared to the US, of course, which is just south of us, but also as compared to the European Union. They're quite aware of that, and they're actually now active members in the ICH. And then the PICS, it's the Pharmaceutical Inspection Cooperation Scheme, is a little bit less important these days. It was a bit more important 20 years ago, so it's kind of being phased out and replaced more by ICH and Health Canada's own guidance. So what does GMP regulate? What areas does it regulate? You can kind of organize its contents into the famous 4 Ps: places, people, processes, and products. So they govern your premises, which means your facility, which houses in which you produce your radiopharmaceuticals. They regulate the equipment that you use to manufacture it and to test them. They regulate the people, the personnel who operate your equipment, who work, who manufacture your drugs, but also in particular the quality control department. This is very important to Health Canada. There's a lot of emphasis in that chapter. In terms of the processes, they regulate your sanitation practices. So this is the cleaning and disinfection of your place, manufacturing, obviously, that's clear, and also the process you have in place to keep records of what you're doing. In terms of the product, obviously, it's the raw material testing that is important, also the packaging material, your actual finished product testing, of course, and samples and stability. So, if you open one of those guidelines or download them as PDFs, they work in clauses. So premises is always the first one, and the clause is very simple. It just says your establishment should be designed and constructed in a way that promotes cleanliness, orderliness, permits effective cleaning, and prevents contamination. Sounds great. Doesn't tell you overly much of what exactly they expect, really. And so what they do then is beneath that, they list interpretations. And the interpretations are really the kind of nitty-gritty details that they look at and that you need to satisfy. So in the example of the premises, for example, they say, okay, you need to have smooth, cleanable surfaces. There shouldn't be any cracks or dents in it because you can't clean them. They said you should not have sharp corners. And if anyone has ever tried to clean out the bathroom corner with the tiles, you know, where the walls came down, there's always some bit of grime there. They don't want to see that. So they specify coved floors that are easily cleanable. They also say that you should design and implement your layout in your radiopharmacy to minimize movement, because personnel shed particulates, they shed bacteria, they shed viruses, as now we all know after COVID. So you need to minimize that. So it needs to have a logic workflow through your department. They're big on that. They specify, for example, that in certain areas you should have no drain or sinks because P-traps are not a clean environment. They also define what type of air filtering you should use in your facility, what type of differential pressure and pressure gradients you should have throughout your facility, etc. So the interpretations are really what you should look out for. And so now, instead of going through each of the chapters and telling you what they look out for and rattling it off, that will put you to sleep and I won't get through it. So what I've done instead is I picked an example and which I hope that you're all familiar with. And I want to tell you what each chapter kind of means for this one example. So in that case, I picked the radioactive assay, which is really measuring your multi-dose vial, your patient syringe, whatever it is, in a dose calibrator to know how much activity you have, you're injecting, whatever. And so these are basically on the left-hand side, you can see the headers of the chapters of the regulation. And so on the premises front, the consequence is that they want you to install it correctly at a suitable location. So what is a suitable location? It should be where, for example, for a dose calibrator, the background is low. There's not tons of people walking by carrying activities and not great shielded pots so that your background fluctuates and gives you a wrong reading. It should not be tucked away into a corner such that only people with very, very long arms can reach in and clean it properly, that type of thing. On the equipment chapter, obviously, there's a lot about the dose calibrator, not surprisingly. They tell you that for all equipment that is used in critical steps or for important tests, you should have performed an IQ, OQ, PQ. What is that? It's installation qualification, operational qualification, and performance qualification. So installation qualification is just what it sounds like. You look at what the manufacturer tells you, what it needs. So should be at between 15 and 25 degrees Celsius, should be plugged into a 110 volt outlet, should have a relative humidity of no more than 70%. I'm just making numbers up, but you get the gist of it. So you just kind of look at that, you follow a checklist, you make sure that you install it properly. That's all. The operational qualification is meant as a test that the equipment operates in the way that it was intended by the manufacturer of the equipment. So in that case, this would usually mean that you turn it on, you ensure that it responds to the prompts on the screen, like if you press measure background, that it measures the background, that if you put in a source, a sealed source of a certain activity, that it shows that, etc., etc. The performance qualification adds two variables, and that's, first of all, it adds your operator. So this is no longer the customer support from the company that installs it. So this is your operators, and it adds your sample with your let's call it testing method. So that would be, is my operator able to measure accurately a sample that is contained in my syringe, something like that. You only do that once initially, and you have to redo it every time that you make any major changes, you relocate the equipment to another location or anything like that. So choose your location well initially and hopefully be done with it. I think I'm stuck again. I'm so sorry. Hang on. Let me stop sharing and reshare. It's my laptop every time I'm on a certain thing. Okay, can you still see me? Hello?
We see you, but not your presentation.
Okay, thank you. So let me go there. Okay. Okay, there we go. Now I think I'm back. Yes. Yes. Okay.
You're good.
Okay, thank you. It's every time I get a meeting invite or something. Okay, so the equipment chapter also tells you that you should have written procedures for the use, maintenance, and cleaning of your equipment. I think we all know that. It suggests or strongly urges you to have a prominent sticker on your equipment to display calibration status. So that is to prevent a user from using an equipment that is out of calibration. They will suggest that you have an equipment binder where all the calibration certificates, the IQ, OQ, PQ protocols, the completed and signed off ones are there, and where you have also usage logs. So who uses the equipment? What do they use it for? Were there any special things? You know, was the background shaky or something like that? Any observations? The personnel chapter of the GMP regulations will tell you that you need to train all of your users into the proper use of the equipment, and you need to keep a record. Remember, from a regulatory perspective, if you don't keep a record of something, you never did it. So please keep a record of all the training and file it. They will also tell you that you need to define a training program. So for example, is one initial training good enough for that forever? Can someone be trained once and work for 20 years? What happens if an employee is gone on a lengthy leave of absence and returns after a year of not using it? Do they need to do it again or not? So they want you to have a defined training program somewhere written and then follow it obviously and document whatever training is specified in there. In terms of the quality control department, obviously they say that the quality control department needs to have oversight and sign off on important things. These can be the calibrations, these can be test results for a final drug product, etc. So, and that is a review, it's a second pair of eyes, it's to make sure that what the operator thinks was measured is actually accurate and true. In terms of sanitation, I just started a new slide because it was getting crowded. It will tell you that you need to have appropriate cleaning procedures and again document that appropriately. And I mean, obviously in the terms of those calibrators, there's not that much, it's wiping the screen or something like that. But in terms of general sanitation, they will ask you to define exactly what cleaning products or disinfection products you allow, sporicide, whatever, IPA, what the contact times are. You know, you shouldn't kind of like squish around, but do horizontal strokes or vertical strokes, that kind of stuff. So detailed instructions on how to keep everything clean and sanitized. The manufacturing control chapter would tell you for the dose calibrator that you should include all manufacturing data in the batch record. So if you use, for example, the dose calibrator to define your initial starting activity and that and your end activity, and that you use that to calculate your radiochemical yield, and that is one of your criteria, then you need to input that. In terms of records, you need to keep all the records that you generate for a suitable time. And I know that suitable time is not very precise. It really depends on which of the regulatory pathways you follow. If you are a manufacturer of an approved FDG product, for example, it's 1 year after expiry of the batch that you used to produce, you can get rid of documentation. However, if you are doing another drug and you use a CTA, you are required to keep all documentation for 25 years. So you need to check out the regulations, and then depending on whatever activity you're doing, choose the applicable time and document that as well, so that your personnel knows what to keep and what to throw out. Raw material testing obviously doesn't really apply in a dose calibrator, but the packaging material applies. So they say you have to use approved vendors. An approved vendor or a vendor qualification program for the vials or syringes that you're using to measure your radiopharmaceutical in there means that you define who you can purchase it from, that you are reasonably certain that it's a high quality vendor. You will not kind of adulterate a product or repackage, or, you know, like put a new expiry sticker on top of the old expiry date or something like that. And that you are aware if your supplier is changing something in the product that could have an effect on the quality of the product, and hence then an effect on the quality of your own radiopharmaceutical, that they would let you know. So your vendors need to have some form of quality program in place as well. They also want you to have, for all the materials that you're using, and that is true for raw materials, packaging, etc., have a specification. So, for example, the vial, what type of glass does it have to be? What is the rubber septum made of? Or if you have a syringe, what type of body is it? You know, does it have a rubber seal at the bottom, or that kind of information? They want you to test it when it arrives on site, a shipment, and then have a formal release procedure prior to it being used in the manufacturing. So this is basically a material control program that the regulations mandate you have in place. I'll skip the finished product testing because there's a lot to say about that, and I'll go straight to the samples. The guidance that is specific for radiopharmaceuticals say that you do not need to keep radioactive samples because as they decay, they become kind of useless. But for vials or syringes or something like that, you do have to keep retention samples, and of course also for your finished product. And you have to keep sufficient product material or sample material that allows you to redo tests should it become necessary because something doesn't quite add up or you need to do an investigation. Stability doesn't really apply in the terms of the radioactive assay. In general, of course, we all know that under GMP regulations, you need to test the stability of your radiopharmaceutical in its container. So that could be the glass vial or a syringe if you ship out syringes, for the time that you put as expiry date on the label. And now let's go to finished product testing, because that is obviously the most important one for the radioactive assay. You need to have a written test procedure, goes without saying. You need to have a pre-test system suitability before you use it, or at least once a day before you start using the equipment for the day, to ensure that the equipment is working appropriately. In terms of those calibrators, these days, this is usually already figured in, it's programmed in the software. It's called the daily test, where you measure a constancy and accuracy source or something of that nature. You, of course, need to have written acceptance criteria for your finished product. So in terms of the dose calibrator, that would say, okay, my FDG lot needs to be, you know, no less than 10 gigabecquerels, not more than 25 gigabecquerels, or whatever the numbers are. I'm just making them up. And your SOP should have instructions on what to do when they're not met. So you should say, okay, if you're outside of that range, please inform the quality control department and ask them to initiate an out of specification investigation for that. You need to document your test results. So that means if you have a printout, that is the preferred way, you should initial and date them. If you don't have a printer, and sometimes depending if you're working in a clean room, a printer might not really be possible because it generates too many particulates, and you just basically transcribe information from a display screen, they usually want a second pair of eyes to kind of countersign initial to make sure that you're recording accurately. They also want a quality control department to perform regular data integrity checks. So if you say print out or generate a PDF as a result of a test, they want the quality control department to go back into the raw data file of the computer on a regular basis and ensure that, you know, like in the process of generating a PDF, either the software or the user didn't change any information and make a test just pass when it was just out or something like that. In general, Health Canada prefers if your test methods follow USP. If you do follow USP, you just need to do a method transfer study. If you don't follow USP, you need to fully validate it for approved drug products and likely for clinical trial phase 3. For BRAPs 1 and 2, you don't need to do that. So what does validation mean? It is defined best, in my opinion, in the ICH guidelines, guideline Q2R1. Again, this is available free of charge on the internet. And it tells you that you need to check for accuracy, precision, specificity, the detection limit, the quantification limit, linearity, and range. So what does that mean for the dose calibrator in particular? It means you do your daily tests for constancy and accuracy, and you start doing that in your operational qualification when you first set up the instrument, and you just do it every single working day. It also tells you that you have to do a linearity test and a geometry test. Geometry test means, for example, if I'm using a 5 ml syringe for a patient dose or something like that, it makes a big difference whether you have a 1-inch needle attached to it, a 2-inch needle or something like that. So you need to test the geometry for every single geometry that you are actually doing later, right? So that can, if you have like a 5 ml vial, a 10 ml vial, a 20 ml vial, a 1 ml syringe, a 5 ml syringe, a 10 ml syringe, you need to do a geometry test for all of those. Good news is you only need to do it once on the dose calibrator, unless you send it out for some major repair or replacement, then you need to repeat that. And the linearity test obviously is the entire range of radioactivity to ensure that you're still in the linear range of your detector. Very quickly, the sterile drugs guidance is where basically most of the really annoying regulations come from, and I mean annoying in the sense that they're kind of onerous and burdensome. So they define room classifications from A to D. They correspond to the ISO 4.8 to ISO 8. D stands for dirtiest, so as you move up in the manufacturing, you go from D towards A. They define that the terminal sterile filtration of your drug product and the dispensing has to happen in a grade A. There are no exceptions to that. Regular manufacturing needs to happen at a minimum at grade C. And the criteria for grade A to D, there are really 3 of them. First of all, in terms of rooms, there's number of air changes per hour, because that tells you how many people, how much material you can bring in, and how quickly it will clean itself up. Obviously, the number of particulates at rest and in operation are measured, and they need to meet preset criteria. So those are set by Health Canada, and you can go lower, but you can't go higher. And it's by the results of environmental monitoring for surface contamination, so that is done by contact plates, and by the air monitoring, and that is done by active air sampling and by settled plates. All of those classifications are really based on the ISO norm 14644-1. The sterile drug chapter also tells you very clearly about how often you have to monitor your clean room for environmental conditions, and what the action limits are. The alert limits, they leave a little bit more up to you, so you can choose those wisely, but the action limits are quite clearly prescribed. They also tell you that you need to have some supervision by a person trained in microbiology to know what they're doing. They define the gowning that you have to wear depending on the room classification, so there's a table in there. They also define the differential pressure between room grades. And this is unfortunately in conflict with CNSC. So CNSC, wanting to protect the worker, wants to have basically the lowest pressure where your radioactive source, in this case your finished product vial is. The Health Canada wants to protect your product, so they want the highest pressure there to keep basically all the particulates that you generate away from the product. Usually the way to solve that is that you have a hot cell that is under negative pressure to the room, but inside the hot cell you have a laminar flow and a negative pressure. So that requires usually some explanation when inspectors come in. Aseptic preparation, and I think I'm running out of time. Yes, I'll finish up. Aseptic preparations, they want you to have media fills, bioburden studies, sterile filter testing, and product sterility testing to ensure that your product is actually sterile. And this is not as easy as it sounds, but that there is not enough time to go into why this is not as easy as it sounds. But especially the product sterility testing is quite a contentious issue in the radiopharmacy world in Canada. And I think that's everything. And I'm open to question, but I think we'll having a question and answer session later. What I'll do is I will stop sharing my screen so I can see people.
Thank you so much, Esther, for this very instructive lecture.